Okay, can you see the screen, students? Please switch on your videos. Yeah, let's start this today. Okay, so in the previous class, are you sure I didn't discuss this? Because I remember discussing the types of seeds. I did? No. Okay. Now see, but this plumule and monocot and dicot I've discussed, right? Okay, so now talking about the seeds. So just like how the mature ovary becomes the fruit, the ovule becomes a seed. And when we talk about the seed, it is mostly a fertilized ovule. Now, seeds are of three types, depending upon whether or not endosperm is left in them. We see that there is endospermic seed, non-endospermic seed, and perispermic seed. Now, endospermic seeds are also called as albuminous seeds because here the endosperm is not completely consumed by the developing embryo. It's not consumed and it is still left in the seed. So when endosperm is found in the seed, we say that that is an endospermic seed, meaning endosperm containing seed, such as wheat, maize, barley, coconut, castor, sunflower. All of these would be the example of the endospermic seed. After that, we have the non-endospermic seed, which are also known as ex-albuminous. Now, non-endospermic means that they will not be having any endosperm because their endosperm has been consumed by the developing embryo. So, this one is found in the case of pea, bean, and groundnut. Lastly, if you people remember nucellus, which was the nutritive tissue found in the ovary or the ovule. So when in the seeds, you find nucellus being persistently present. So that residual or the leftover nucellus is called as perisperm. So those seeds that do not have endosperm as such, but instead of that, they contain the perisperm in them. So we call them as the perispermic seed, such as black pepper and sugar beet or beet. So these are your three different types of seeds depending upon whether or not they have the endosperm. We have this. Now talking about the structure of the seed. So the seed has a tough protective covering, which becomes like, if you remember in the ovule, there was integument, outer integument, inner integument. So those integument, they start and become the coat of the seed. Seed coat is made by them, which is very tough. It is very protective in nature and it doesn't let the seed die. So that is the seed coat, which has two layers. The outer integument becomes the testa and the inner one becomes the tegmen. So testa and tegmen are the names of the two layers of the seed coat. Now you people might have seen that in the seed, there is a tiny small hole present. That is the micropyle, which still persists. Why? Because of the entry of oxygen and water, so that the seed could germinate. Like whenever we put a seed in the soil, we put water. So how can the seed sprout? It's because the water went inside that seed and caused the seed to germinate. You already know endosperm, it may be present, it may be absent, but it is a part of the seed. And then embryo. Embryo is the part that is present very inside, which has this capability to give rise to the new mature plant. Now this was about the seed. Now there are two phenomena associated with the seed, and that is dormancy and seed germination. Both of them are opposites of each other. Dormancy is when the seed will not germinate. Here, the embryo is inactive. The seed is not going to germinate. Why? This is a protective mechanism, meaning whenever there is not enough, you can say, favorable conditions like not enough heat and not enough moisture, not enough oxygen, not the right temperature. So in those cases, the seed is not going to germinate. Why? Because it as a protective measure, it has become inactive. It will not germinate until and unless the favorable conditions come back. So because of the absence of favorable condition, the dormancy happens, and during this time, the moisture content of the seed decreases and it reaches 10 to 15%. Normally, the seed has enough moisture, but during this time, it decreases and the seed becomes inactive. The exact opposite of dormancy is seed germination. Now here, under the favorable conditions of light, temperature, moisture, and everything, the seed is going to germinate and give rise to the seedling, which is like a tiny, tiny plant, right? So that is called as seed germination. Always remember that this will also happen under the adequate moisture. Adequate means the enough amount of moisture, oxygen, and temperature, or else this will also not happen. Now, the ultimate product of reproduction in the case of plants is the seed, because you know that seed only will give rise to the new plant furthermore. So let us see the advantages of the seed to angiosperm. So seed, they have better adaptive strategy because of the presence of seed coat, dormancy phenomena, even if they are dispersed elsewhere and they do not have enough favorable conditions. So they'll either stay inactive. If they get the condition, they'll become active. So seed has that adaptive strategy. Then, because of the presence of endosperm, they have a lot of food reserve for them. It's like a pantry. They have a food reserve so that wherever they'll, whenever they are to, give rise to a new seedling or they are to germinate, they'll have enough food reserve in them. Then seed coat provides them with the protection to the young embryo. And lastly, because seed is you can say it is a product of sexual reproduction. So obviously there are going to be variations in them. Variation means introduction of new characters, which helps them to survive better. So that is your seed. Seed is basically they are advantages because of these 4 points. Clear? Any doubts? Oh. Now, the seeds, they are very important for agriculture because they are showing 2 properties, or they have 2 properties. First property is dehydration. Now, seeds can be stored if you dehydrate them, right? Drying of the seeds to maintain the storage life. And second, the dormancy that we talked about. Because of these 2 properties, seeds are very important for agriculture, for long-term agriculture, like for storage and for seasons and everything. Then, just like how, if you remember, we studied pollen viability, same way there's seed viability. So this is the time period for which the seed is, you can say they'll have that power, they'll have that ability to germinate. If the seed loses the ability to germinate, we'll say that the viability is completed, it's over. So oxalis, here the seeds, they lose the viability within few months. They'll not be active after few months. Lupine, the scientific name for which is Lupinus arcticus. This is the oldest seed which was excavated in the Arctic tundra. And even after 10,000 years of dormancy, it germinated. Imagine such a long viability it has. Then the date palm, called as the Phoenix dactylifera. This was excavated at the King Herod's palace near the Dead Sea, and this was also viable for 2,000 years. So, meaning even after 2,000 years of dormancy, it germinated. Then the number of seeds in a fruit will always be equal to the number of ovule in the ovary, because you people know that ovule only becomes the seed. So, however many ovules are going to be there, that many seeds are. But always remember, the number of ovules can never exceed the number of ovaries, isn't it? However many fruits are going to be there, same number of seeds are going to be there, just like that. Now, the orchid fruit contain thousands of tiny seeds in them. There are certain parasitic species that also contain many tiny seeds, such as orobranchi and striga. Now, after the seed part is completed, we move on to the last part of the fruit, last part of the reproductive series, and that is the fruit. So fruit is when the ovary becomes ripened, it forms a fruit. So the ripened ovary is a fruit. The wall of the ovary becomes the wall of the fruit and it is called as pericarp. Now, depending upon their origin, the fruits are of three types: true fruit, false fruit, and parthenocarpic fruit. Now, you know that the fruits, they are developing from ovary in normal case scenario. So, all those fruits that develop from ovaries, they are called as true fruit. They are the real fruits, such as mango. Second, if the fruit develops from the parts other than ovary, like thalamus. So that is false fruit, meaning it's not developing from ovary, but it is a fruit, such as apple, strawberry, cashew. And then comes the parthenocarpic fruit, wherein the fruit develops without fertilization. It is developed artificially by using certain hormones. When you apply hormones to the plant, it can induce flowering, it can induce fruiting and seed formation. So when the fruit is formed without fertilization, we call it as parthenocarpic fruit. The method of forming this fruit is called as parthenocarpy. Clear? One such example is banana. And the two hormones that are used are auxins and cytokinins. These are the two plant hormones that are utilized to make the seedless fruit or the fruits without fertilization. Now, why am I saying they are seedless? Because since the fertilization did not take place, how will the ovule become the seed? So here they do not even have the seed, such as banana. Clear? Now the fruits, they can be fleshy or they can be dry. Now the fleshy fruits such as mango, guava, oranges, and dry fruits we have mustard, groundnut, meaning they do not have a sticky mesocarp or the sticky edible portion. Coming on to the next few topics, that is apomixis and polyembryony. Now you saw how the formation of fruit without fertilization was called as parthenocarpy. Same way, when the seeds are formed without fertilization, we call it as apomixis. It's not seen in every plant. It is most commonly seen in grasses and the members of Asteraceae family. So it is a phenomena which is basically an asexual reproduction, because here fertilization or gametes are not involved, but it is mimicking sexual reproduction. Now, apomixis is of various types such as adventive apomixis. Here, the embryo is going to develop from the sporophytic cell, which is diploid in nature, meaning nucellus, integument, that is going to form the seed instead of the egg fertilized like embryo and stuff forming the seed. So we call it as adventive apomixis, which is found in usually citrus, apuntia, and mango. I think this is the last topic, that is polyembryony. Now, poly means multiple and embryony refers to the embryo existence. So when more than one embryo is found in the seed, we call it as polyembryony. Such a thing exists in the citrus. Now, polyembryony is also one type, that is apomictic polyembryony, meaning this is a method in which you can develop the seeds that are apomictic in nature, meaning they do not need fertilization, and you can have multiple embryo in them. This will make a higher yield plant which has stress resistance as well. But again, these are expensive as far as the large or mass scale production is concerned. But apomixis, meaning making the plants without fertilization, is actually a solution to most of the problems. Why? Because it is quicker. And the process of fertilization will take so much of time. First, the gamete will be formed, they will be transferred, meaning pollination will take place, then fertilization will take place, then the development will take place, right? But apomixis is like a quick and easy fix. Clear? Any doubts? So with this chapter is done for.
In this,
Huh?
the embryo will be there.
In this,
In this apomixis, embryos will be present.
yeah, embryo is going to be present, but you can say it is the seed only that you're forming, but without fertilization. Meaning you're using artificial means to make this. Understood? So yes, it's going to be there, but the origin will not be sexual reproduction. Clear?
Yes.
Yeah. Now tell me, students, what was the last thing you people were writing?
Megasporogenesis.
Megasporogenesis, that far. Oh God. Megasporogenesis is like the first PDF. Achha, see, I know this completing this PDF is not possible in today's class, obviously. So, like you write for a little while, till then I'll rest my throat. Then we'll start a new chapter today. Okay. And these notes I'll send you in the form of PDF. But again, you people will not get this PDF printed out. You will write it down. Clear? You will leave pages and you note it down. Because if you take out PDF, trust me, I am and I have been a student as well. We never open PDFs. We never open the prints also. So you will write it down. This is where you were writing, students.
Yes, ma'am.
So you note it down. After a few minutes, we'll start a new chapter today.
Done, ma'am.
Both of you are done?
Yes, ma'am. Done, ma'am.
Done, ma'am.
Ma'am, can you scroll a bit down? Done.
Ma'am, could you please scroll a little up?
Could you scroll a bit?
So if you are done till here, the development of female gametophyte is done.
Yes, ma'am. Done, ma'am.
Yes, both of you are done, students.
Yes, ma'am.
So now we're going to begin with this chapter, that is the second chapter, human reproduction. Now again, this chapter is, it contains every single thing as far as the human reproduction is concerned. We're going to see the various, first we're going to see again the pre-fertilization stages, the fertilization stages, and ultimately the post-fertilization stages here. So when we talk about human reproduction, so obviously Homo sapiens are involved in this. Now the various things that we're going to see in this chapter are, the first one is gametogenesis, which is again formation of the gametes. Then this gametogenesis we're going to see in the case of male, the case of female. Then insemination, which is the transfer of gametes in the human case. Fertilization, implantation, gestation, and ultimately parturition, which is the delivery of the baby. But before we move on to all of this, we have to study about the male and the female reproductive parts. So first comes the male reproductive system. Now, male reproductive system is present in the pelvic region of the males. And what are the constituents here? There is a pair of testes, which is basically the primary sex organ. What is a primary sex organ? A primary sex organ is the organ that will be responsible for the formation of the gametes as well as it will be responsible for the secretion, like formation and secretion of hormones. Then in the male reproductive system, accessory ducts are present, which are basically responsible for the transferring of gametes. Then there are glands that are associated with reproduction. And lastly, the external genitalia. Now, when we talk about the male reproductive system, it is present in the pelvic region of the males. And talking about the testes being the primary sex organ, so see, this is the picture of the human male reproductive system, wherein this is where the testes are. Now, the thing is that the testes being the sex organ or the primary sex organ, they are not found inside the body. Rather, they are found outside the body. And they are situated outside the abdominal cavity, inside a pouch, like structure called as the scrotum. But the question comes along, why? Why is it situated outside? There must be some reason. So can someone tell me what is the normal body temperature of human beings? What is the normal human body temperature? 37 degrees Celsius. Okay, it is 37 degrees Celsius. Now, for the formation of gametes, male gametes, like 2 to 3 degree lower temperature is required as compared to the body temperature. So how would that be possible inside the body? So that is why the testes, they are situated outside the body to help to maintain the low temperature. Now, this low temperature is extremely important as far as the formation of sperm is concerned, without which the sperm will not be formed. Now, in an adult, each testis is usually oval in shape and the length of the testes is 4 to 5 centimeter. The width is 2 to 3 centimeter, width meaning thickness. Now, the testis is covered by a dense protective covering as well. Now, talking about the ultrastructure. Now, see, inside each of the testes, there are 250 compartments. These are the checkered boxes that I've made. These are the compartments inside the testes. And how many of them are there? 250 compartments are there. Okay. These compartments are called as testicular lobules. Okay, testicular lobules. Now, inside of each of these 250 compartments, there is the presence of 1 to 3 highly coiled seminiferous tubules. So, like for example, if inside this there are 1 to 3 highly coiled seminiferous tubules in each one of them. Understood? Now, now we are going to see the ultrastructure of seminiferous tubules. Now, when we move like completely inside the seminiferous tubule, on the inside, it is lined by two types of cell. The first type of cell is male germ cell, which is also called as spermatogonia. And the second type of cells are called as Sertoli cells. Is this clear, students, up until now? Am I going too fast? Wherever you feel like I'm going too fast, tell me. Now, the male germ cells, obviously, now see, in general, people have to understand and remember forever, that the process by which the gametes is formed is called as meiosis, right? Which is the reductional division. And apart from that, can you all tell me what is haploid and diploid quickly? What is haploid? This is the basic. This so you have to know any way or the other. Like see, haploid, diploid, so you people have studied in sexual reproduction also. So you should be able to answer. What is haploid and diploid? Like a cell is haploid. What do I mean? Don't remember? Anisa, what about you?
Ma'am,
What is
haploid is like when there is only one set of chromosomes, and
any
diploid is two sets of chromosomes.
any cell is called as a haploid cell when it only has one single set of chromosomes. Acha, how many chromosomes do us human beings have?
23.
23 pairs of chromosomes. Okay, meaning all in all, we have 46 chromosomes in our body. But when 46 is the diploid number, meaning 23 is one set, another 23, together we have 46. But when you talk about a haploid cell, it would have only 23 chromosomes. Okay, so haploid and diploid things should always be clear to you. Now, furthermore, when we talk about the male germ cells, so they are like basically the male germ cells that are found inside the seminiferous tubules, they are going to form the sperms, which is the male gamete, human male gamete, by the process of meiosis. And the Sertoli cells, they will provide nutrition to the sperms. Sperms are also called as germ cells, meaning the reproductive cells are also called as germ cells. So these are the two basic functions. Now, the region that is present outside the seminiferous tubule, we call it as interstitial space. It's like a space present outside the seminiferous tubule. This space has blood vessels, it has interstitial cells called as the Leydig cells. Now, the male hormone that I was talking to you about, that male hormone is synthesized by these Leydig cells. What is the name of that hormone? It is called as androgen. Apart from that, here other immunologically competent cells are also present. Is this much clear, students? So this is like sort of like a revision that people should, know that the number of chromosome or the number of sets of chromosome is called as ploid. When it is set, we call it as diploidy. When it is a single set, we call it as haploid. Now positions also you people should know that dorsal and posterior, these two names, they refer to the back position. Ventral and anterior, these two names refer to the front position. Lateral means sideways and apical means the tip portion. Okay, these are the names in terms of biology which you people should. Is this much clear to everyone?
Yes.
Any doubt?
No.
So this was about the male reproductive system's first part, that was the accessory duct. Now after that we come on, not the accessory duct, the primary sex organ. After that we move on to the accessory ducts that are found in male, human male. So there's brachydestis, there's vas efferentia, there's epididymis and vas deferens. These are the different names of the male accessory duct. How does it begin? See, the process begins like seminiferous tubule. They are going to open into the retitestis, which is going to open into the vas efferentia, which will open into the epididymis, then to the vas deferens, to the ejaculatory duct, and ultimately to the urethra. Okay, this is the pathway. Like when the sperms will be released out of the body, this is what the pathway is that it will be following. Clear? This coiled structure that is found, we have the seminiferous tubule. Now, seminiferous tubule is going to open into the retitestis, which will open into vas efferentia, which will open into the epididymis, this part, and then it will open into the vas deferens, which will move towards and it will combine with the urethral duct, and then be, like the pathway is going to be in the urethra. This is how the pathway of sperm looks like. And the sperm is released outside the body with obviously the help of the accessory ducts. If they are not going to be there, then the sperm will not be transported out. Is it clear, students?
Yes.
Okay. So that was the pathway that you people must remember many a times. They'll ask you the correct sequence. So you should know what comes after what. Now, the seminiferous, this the same exact thing I've written in the language. So the seminiferous tubule, they open into the vas efferentia through the retitestis. The vas efferentia leaves the testes and open into the epididymis. Where is it situated? It is situated posteriorly at the posterior surface of each testicle, meaning at the back side of the testicle. Then they're going to open into the vas deferens. Now, this epididymis is going to receive ducts from the glands. Okay, the different male reproductive glands that are there, they will be secreting their secretions into there. Then the external opening of the penis. Now, see, the penis is the external genitalia in the human males. Okay, it is the genital organ that is externally present. And the opening of the penis is called as the urethral meatus, and it is made up of special tissues that help in the erection process. Erection means the blood will flow towards it and it will get harder in, you can say, firmness. That's what erection is. So this is the basic structure of the human male reproductive system. Now, the next thing that we have are the male accessory glands. But up until now, is it clear to both of you students? Actually, remember one thing I'd like to tell you, that since I understand that this is, human reproduction topic, and, like you people have already studied this in class 10, but now in 12th, you have it much more vast. It's like in-depth knowledge about everything, at terms of the cellular level, the structural level and everything. So in this chapter, I'd ask you people not to feel hesitant to ask your doubts. Even if you're asking about a reproductive system or anything of that, but this is your chapter, this is your course, right? So no shyness and no feeling awkward. If you have any doubts anywhere, you can ask. Understood? It's not that only I'll speak, I'll teach. And out of shyness, you'll not ask me. That shouldn't be the case, because you will be asked a lot of questions from this chapter in your examinations. So you must pay attention. And despite that, if you have any doubts, feel free to ask, okay? Now, coming on to the male accessory glands. Now, accessory glands, why are they named so? They are named so because they're not involved directly in reproduction, but they play a major role indirectly. They form secretions and fluids and nutrients and everything. So there are three, basically, the reproductive glands. There is seminal vesicle, the first one. There is prostate gland, the second one. And then there is the bulbourethral gland. So this picture shows you the position of the various glands, and this is like a lateral view of the picture. So it shows you where is it situated. So these three we're going to study: seminal vesicle, prostate gland, and bulbourethral gland. These three glands are the male glands, right? These are the male accessory glands. Now, the first one is the seminal vesicle. Now, these are a paired gland. Paired means how many of them? Two of them will be present, right? It is present in a pair, and it is tubular and coiled gland. It is tube-like and it is coiled. So where is it situated? It is, now the positions are very important. So where is it situated? It is situated behind the urinary bladder. So this is this pink portion is the urinary bladder, pink name. Each portion is the urinary bladder, and behind it is the presence of seminal vesicle. Behind. Now what does it do? It secretes a viscous fluid. Viscous means thick fluid, which is the main part of the ejaculate. What is an ejaculate? It is the release of sperm. Okay, and the sperm is not just like that released out. It is released with some sort of a fluid component to it, and that is made by the seminal vesicle. Apart from that, the seminal vesicle also provides fructose, which is a sugar, the ejaculate. Why? So that the sperms can have enough nourishment. That's the case. The second accessory glands of human males are prostate gland. Now, this gland is a chestnut-shaped gland which is situated at the base of the bladder, not behind the bladder, at the base of the base. Understood? So this is present at the base, and it is a chestnut-shaped gland. What does it do? It provides an alkaline, which is a basic component to the semen, not acidic in nature, basic in nature. Apart from that, it provides the calcium ion, phosphate ion for the nourishment of the sperm. Now, you might have heard like one of the type of cancer is the prostate cancer in males, right? That is the cancer of this gland. When this gland starts secreting or, starts having a tumor or stuff or anything of that sort. Many a times in many males, this prostate gland can get swollen up or can get inflamed. So the inflammation of the prostate gland is what we call as prostatitis. This is something extra that I've added. It is not in NCERT, but you should know that any word that ends with the suffix itis, bronchitis, prostatitis, okay, then you have rhinitis. So any word that ends with this suffix itis refers to inflammation. Inflammation means swelling, redness, and all that. Always redness. Now, furthermore, the next gland, the last gland of human male is the bulbourethral gland, which is also called as the Cowper's gland. Okay, it is a pea-sized gland which is paired, meaning two of them. Prostate gland is not paired, seminal vesicle is paired, and bulbourethral is paired. So, it is situated adjacent to the urethra at the base of the penis. Now, see, adjacent. This is the urinary bladder. This is the bulbourethral gland, which is situated at the base of the penis. So it secretes the mucus to act like a lubricant. So the function of like the human male reproductive system is completed now, Have you people understood the structure, the functions, and different parts of it?
Yes, ma'am.
Any doubts? Anything you want to ask me? No. All right. Okay. Now let's get back to the notes writing. So we have started this chapter today, human reproduction. Now you just have, I think, 10 minutes left to the class to get over. So I am going to send you the sexual reproduction in flowering plants notes. Okay. So you can copy down the rest of it, but till the class gets over, you can copy down your notes. Is my screen visible to you all?
Yes, ma'am.
Now you note down. Done, students?
Ma'am, 1 minute.
Right, students, so let's just keep